Stem Cell Research by Jolie House, Jen Kaflewski, Brittany Lee, and Hannah Mumber.
Who knows about stem cell research? I knows!
Houston, we have a problem. Some people don't know about stem cell research. Well, good thing it isn't rocket science. This is Hannah speaking, and I'll introduce stem cell research. First, the National Institute of Health defines a stem cell as an unspecialized cell capable of renewing itself through cell division, sometimes after long periods of inactivity. Under certain physiological or experimental conditions, it can be induced to become a tissue or organ-specific cell with a special function. Stem cells work as a repair system, and they continue replenishing cells in the body as long as the person is alive. There are two main kinds of stem cells from animals and humans: embryonic stem cells, those coming from an embryo, and non-embryonic somatic or adult stem cells. So that is a basic introduction to what stem cells are. Later on in our presentation, we will discuss some recent research applications, such as how acid baths turn body cells into stem cells for tissue repair, cloning such as SCNT, or therapeutic cloning, and parthogenesis, which is a method of scientifically induced reproduction. Stem cell research is significant because it increases scientific understanding, improves medical developments, and overall helps to advance society. First, it offers new potentials for treating diseases such as diabetes and heart disease, and several other diseases as well. Its application has been called regenerative or reparative medicine, and tissue engineering is used to create artificial organs to replace defective ones. For example, progenitor cells are used to make artificial bladders. Furthermore, stem cell research increases the understanding of the causes of birth defects. There's also the possibility of generating patient-specific stem cell lines that aid in the study of the mechanism of different diseases in the laboratory. Another benefit of stem cell research is drug development. It enables the creation of models for drug discovery and allows drugs to be tested to see if they have any toxic or other harmful effects. Finally, stem cell research benefits artificial pregnancy. In vitro fertilization and parthenogenesis are some techniques utilized to help infertile couples have children. Next, we will discuss some of the methods of stem cell research.
Hey everybody, this is Jolie speaking now. The most common method of obtaining stem cells is using adult stem cells that can be removed from adult tissues with little harmful effect on the individual, while embryonic stem cells are derived from multicellular embryos that have been cultured in the laboratory. Here's a list of the current and potential sources of stem cells. Please note that an SCNT is a procedure that bypasses the normal fertilization process by taking the genetic material from a cell in an adult's body and fusing it with an empty egg cell. This is a form of therapeutic cloning, which would allow cells to be customized for each individual and thereby minimize the chances of tissue rejection. Also know that adult tissues are used in bone marrow. Advocates for human embryonic stem cell research want to use embryos that have been frozen at fertility clinics as the main source of embryonic stem cells. When a woman gets pregnant with in vitro fertilization, which is manually combining an egg and a sperm in a laboratory dish by physically placing an embryo in its uterus, not all of the embryos are implanted, and fertility clinics allow these couples to destroy them, donate them to another couple, or freeze them in case they want to give birth to another child at a later time. They use these leftover cells from these situations for research. Parthenogenesis is another method of reproduction. Basically what happens is that a female can generate offspring without the contribution of a male, and this creates a viable embryo. These embryonic stem cells can then be generated. However, this does alter the gene expression on these cells that are imprinted, which can lead to cancer and/or poor growth of tissues. Unfortunately, this method has not been able to generate cells for medical use. A surprising study has found that a simple acid bath might turn cells in the body into stem cells that could one day be used for tissue repair and other medical treatments. The technique, performed only with cells from mice, might turn out to be a quicker and easier source of multipurpose stem cells than methods now in use. Certain stem cells can easily be grown in the laboratory and can turn into any type of cell in the body, which is called pluripotency. Researchers think that these stem cells may one day be used to repair damaged cells in the organs of the body, though these experiments are in the early stages. The researchers tried various types of stresses, including squeezing the cells, but they found that bathing the cells for a half hour in a mildly acidic solution seemed to work best. Though many cells did die from the exposure to acid, among those that did survive, many were glowing green by the seventh day. The researchers called these STAP cells, standing for Stimulus-Triggered Acquisition of Pluripotency. These stop cells, under the right conditions, can form material for the placenta, not just the embryo, which might allow an animal to be cloned just by putting some of its stop cells in a uterus. Fun fact, there's speculation that this might help explain why stress appeared to increase the risk of cancer.
This is Jen speaking now. Part of the reason stem cells are used is because they can proliferate. Proliferation means it can replicate many times, and what makes them so effective is that they can proliferate for over a year in a lab without differentiating into different cells. Embryonic stem cells are pluripotent, meaning they can differentiate into all cell types of the body, while adult stem cells differentiate into cells of their tissues of origin only. Like Jolie mentioned earlier, stem cells are used in vitro fertilization. Because embryonic stem cells are the ones that differentiate into all cell types, scientists have developed induced pluripotent stem cells, or iPSCs. Here, the adult cells are reprogrammed so that they can enter an embryonic stem cell-like stage. These cells are specific to the donor, and therefore it can increase the chance of compatibility. One of the huge results of stem cell research is cell-based therapy and regenerative medicine. What happens here is stem cells are induced so that they can differentiate into the specific cell type needed to repair any damage, like for example in the heart muscle. Stem cells are also used in transplants, and they help treat Parkinson's, diabetes, spinal injuries, muscular dystrophy, and a bunch of other things. And they also are used to test new drugs for safety. Embryonic stem cells can be produced through cloning by using human skin cells and egg cells, and this can lower the risk of rejection in transplants. Also, some animal clones have arisen because of these stem cells, and research with that is continuing on. Here are just some specific examples of stem cell science. For example, it's used to generate healthy heart muscle cells in the lab, and then what they do is they transplant those cells into patients with chronic heart disease in order to repair that damaged tissue. Also in diabetes, what they do is they take human embryonic stem cells and they use these to make insulin producing cells that can later be used in transplant therapy for people with diabetes. And also something I found quite interesting is that they actually implanted deaf adult gerbils with human embryonic stem cells from the early ear cells, and because of that their brains registered sound, and so now they could hear. And what it does is the stem cells restore the dead auditory neurons in their ear, and what they're trying to do now is trying to use it to treat deafness in humans.
Hi, this is Brittany. There are many pros to stem cell research. Stem cells can cure the most traumatic injuries and diseases, including birth defects, spinal cord and organ damage, stroke, diabetes, cancer, and heart disease. Embryonic stem cells can transform into any cell in the body, and they have a very low rejection rate. Stem cells can also replace or repair damaged organs. Stem cells are cells that have not yet differentiated. They have the ability to divide and make an indefinite number of copies of themselves. Stem cells, if retrieved from the umbilical cord, have many cell types, so matching is always possible. It's really easy to donate and store cord cells. Stem cells can create a stem cell line, which is replicating cells from a single blastocyst, and they can be kept alive indefinitely. They also help researchers gain more knowledge as to how cells develop. This gives more insight as to how to treat diseases as various stages of the disease progress. is the potential to carry out experiments for new medicines on stem cells instead of using animal subjects. Stem cell research also has many cons, such as ethical issues. Some people believe that stem cell research is creating life to destroy life, and that people should not mess with God's creations. Even though the blastocyst is 100 cells, it is a stage of the embryo before the cells start to differentiate. Some believe this is murder, even though the blastocyst has no human features. Some believe that more research into stem cells will give humans greater insight as to how to clone a human being, which has many ramifications and ethical issues. And many people believe that the embryo is not right to kill the embryo, which must be destroyed for research, but it is fertilized in a laboratory.
Overall, we are in favor of stem cell research and support its application. Ultimately, the pros outweigh the cons. While stem cell research has been called unnatural, we believe that its benefits warrant its use. As society continues to become increasingly more complex, new technologies are produced with a purpose to help advance society. Stem cell research is one such technology. In our opinion, it would be a shame to let such a potential go to waste.
So here's our bibliography, and I hope you enjoy the wonderful picture of Mrs. Hill from back in '07.